important one for us to discuss, because one of the things we're learning in lab is how to recognize different species of plants, but we need to kind of understand what that term species means. And I know you've probably had some of this in previous classes, because I know I talk about this in one of my chapters for Biology 130, so hopefully some of this will be a little bit of review. We're going to try and expand on a little bit as well, though. Before we get into the concepts themselves, and there's only one that I'm really going to focus on that much, we need to explore some terminology that I'll be using throughout this lecture that I'm going to assume that you're going to have an understanding of after you go through it here. But that way I can just keep using that term and you don't necessarily have to keep having me define that. The first thing we're talking about is something called self-fertilization. This is a situation where you have union of gametes, like sperm and an egg, that came from the same individual fertilizing each other. Okay? So you could have exactly the same flower and sperm from this plant or this part of the plant right or this flower fertilizing the egg in the female reproductive parts of that same flower. That would be self-fertilization. Yes, some plants can do this. They can fertilize themselves. It's kind of the ultimate method of inbreeding, if you think about it, because you can't be any more related to yourself than yourself. Okay, so that's kind of something that can actually lead over the long term to some potential problems in populations if they do nothing but self-fertilization. Alternatively, you can have something called cross-pollination. Cross-pollination is the transfer of pollen from one individual to the female reproductive parts of a second individual. And you're actually going to a different plant, different flower, and you're not fertilizing yourself, you're fertilizing another individual. Which is generally speaking what we want to do. Now, is that other individual the same species as you? You're hoping so, but it doesn't necessarily have to be. You're just going to a different individual. Now, if you end up fertilizing a different species, an individual that's a different species, that's what is referred to as hybridization. Hybridization is sexual reproduction between different species. And yes, it does also happen in plants. We're actually going to talk about how that leads to a lot of complications when it comes to some of the definitions that we use for species. The fact that plants can readily hybridize oftentimes. We also need to be somewhat familiar with the reproductive parts of flowers, because some of the reproductive isolation mechanisms I'm going to talk about are going to relate specifically to flowers. Angiosperms are 95% of the plants that are out there, so it's very highly relevant. We're going to hit this, of course, in lab as well, because we'll have a whole lab that's based on floral morphology where we actually get to dissect flowers and see some of these in more detail underneath the microscope. But let's go through the basics. Again, this should be kind of 130 stuff that we've had before. You have two whorls inside the flower that are reproductive, and two whorls that are not reproductive. Out here on the bottom, you have your sepals, and then next layer up is the petals. Those are non-reproductive whorls because they're not involved in making sperm or egg. But the next whorl in, this one that you see right here, that's our male reproductive structure. And the one in the center is going to be our female reproductive structure. The female reproductive structure is called a carpel or pistil. I will elaborate in more detail later in lab what the difference between those two terms are right now. They are subtly different from each other, but right now we're just going to use them interchangeably. Okay? And the carpel or pistil is broken into different parts that do different things. On the very top right here, you have what's referred to as the stigma, which is usually going to have some sort of a sticky substance on it, because the job of the stigma is to catch the pollen. Okay, so if we've got pollen from another flower coming here, either by wind or by some sort of a pollinator, the hope is that's going to stick onto that stigma right there and stay put. Then you have the long stalk down underneath there. It doesn't have to be this long, but for this one it's long. That's referred to as the style. It's in between the stigma and then the swollen part down here, the ovary. That's where the style is located. And it holds the stigma up. The swollen area down here is referred to as the ovary because it houses the ovules, which are the immature seeds. We may have one, may have many, depending on what species we're looking at. Then the male reproductive structures are referred to as stamen, and they are also broken into two parts. So you have the most important part on the top here, that's referred to as the anther, which is what's responsible for making the pollen. And then you have the stalk that sits on, that's referred to as the filament. Okay? It doesn't make pollen, it just holds the anther up. So stigma, style, ovary, anther, filament. And we'll especially come back to this when we talk about the incompatibility, the style, stigma, and incompatibility in just a little bit. Okay, so what exactly is a species? There are many, many different ways you could actually define a species. And you probably have seen a number of different definitions in different books. There is actually no consistent definition for a species that actually works for every single living organism out there. Okay? What what works for one group of organisms may not necessarily work for another group, and we're gonna talk about why that is. But one of the most common ways to define a species is to use something called the biological species concept. Really popular with animals, because it's easy to test with them. But if you're gonna be the same species, under this definition, you have two individuals, and you're trying to figure out if they're the same species or not. By this definition, those two individuals have to be able to do two things. One, they have to be able to interbreed with each other. That's the first step. That involves mating, fertilization, things like that. That has to be able to happen. And then two, they have to be able to produce viable and fertile offspring. They have to meet all of these criteria right here in order for those two individuals to be considered the same species. You can't just meet one. So if they only are able to do number one, but not number two, they're considered different species by this definition. Now the term for that, if they don't meet both these criteria, is something called reproductive isolation. Because they're essentially reproductively isolated if they can't get to the final step where they have an offspring that can actually reproduce itself. And so by that definition, they would be considered different species. But there's a lot of different ways these steps could be interrupted or stopped. And we're gonna look at some specific examples of each of those as well. And we're gonna categorize them in addition to that. To understand what the categories are, you first have to kind of take a step back and think about what are all the steps that actually happen when an organism sexually reproduces. And what are the steps involved in sexual reproduction? One of the things you're probably finding out, or hopefully at this point kind of realizing, is that this definition is only gonna work if you're an organism that reproduces sexually. It doesn't work for other ones. We'll talk about that in a second. Okay? But let's look at sexual reproduction, the process here, and the steps that are involved to see how we can categorize these different methods of reproductive isolation. And we classify them first by the time at which the effect during sexual reproduction occurs. More specifically, does a zygote form or does it not? Okay? So here are our basic steps of reproduction. First step, you've got to be able to get the egg and the sperm together. I'm not talking about them fusing yet, I'm just bringing them together. Okay? So in the process of plant pollination, that's what we're doing. The pollen comes to the other female reproductive part. That is bringing those two gametes together. They haven't fused yet, they're just being brought together. So we have mating, is what that's referred to. The next step that has to happen in sexual reproduction is that egg and that sperm actually have to fuse with each other in the process of fertilization. When they do that, they make a cell, a single cell that's diploid, that is referred to as the zygote. This is essentially the first cell of the new individual that's being produced. And since all plants are multicellular, we're definitely going beyond this point. And that zygote is going to start to divide and grow and develop. That's our next step. You have to have development. Development is essentially defined as all the series of steps that are involved in going from a simple form to a more complex form that's ultimately able to reproduce. Even we go through development. We're not born with the capabilities of reproducing, and we get bigger and bigger, and we actually have to go through some developmental changes before we can actually reproduce. Other organisms may go through something more extreme, like a butterfly hatching out as a caterpillar, which then goes through several molting stages, including the pupa stage, where it transforms inside and out in terms of its physiology and its anatomy, and becomes the butterfly. Right? So that's all related to development. That's gonna happen. And then eventually, the goal here is for you to get to an adult, but not just an adult, but for that adult to then be able to reproduce. All four of these steps have to occur. The mating and fertilization is that interbreed part of that condition we saw just a second ago. The development is part of that viability of the offspring that we talked about, and the reproductive adult is the fertility of that offspring. Okay? So what are these different categories then for reproductive isolation? Well, you'll notice that in some cases, some of the steps, if you get through them, you can actually get to the zygote, and some of the other ones you may not if you stop them. So here's how we can kind of differentiate the two groups. Does the zygote form or not? What are we preventing from happening? Are we preventing mating from happening, fertilization from happening? If so, we are not even allowing the zygote to form, and we would call those pre-zygotic isolation mechanisms. Are we preventing development from occurring? But the zygote actually was able to form, which means mating and fertilization did happen, right? So development is not occurring, or the adult can't reproduce. Well, now we're beyond the point of the zygote, so we can call those post-zygotic isolation mechanisms. And they're still gonna prevent them from being successful, ultimately making a viable fertile offspring. But they've made it through the first two steps. So at least I can give them credit for that. They made it past the zygote, post-zygotic. But there are specific examples that fall within each of these categories. One of the things I want you to think about as we go through the specific examples is exactly which one of these steps is being stopped, right? Is mating being stopped? Is fertilization being stopped? Is development being stopped? Is the offspring being prevented from reproducing? So one of the things that you can look at for the pre-zygotic mechanisms is that they can occur either before mating occurs or after mating occurs. If they occur before mating occurs, we refer to them as pre-mating pre-zygotic mechanisms. Okay? So we're stopping step number one. They don't even get the egg and sperm together. That step doesn't even happen. In most cases, this is because pollination is not going to be, in plants, is not going to be successful. Right? You can't successfully bring that pollen grain to another flower, then you can't bring the sperm to the egg. But it can still be pre-zygotic and still make it through this step. Right? We refer to those as post-mating pre-zygotic mechanisms. And in this case, we actually make it through this first step. The egg and the sperm are brought together, but for some reason, fertilization is not able to occur. Usually there's something chemistry related going on there. We'll talk about an example of that. So we've got pre-mating mechanisms. It's assumed that you know that they're pre-zygotic as well. And then you have post-mating pre-zygotic. Okay. Now, post-zygotic mechanisms. Mating and fertilization occur, but something's going on beyond this point. We're gonna have some sort of an issue with the offspring. Either they're not gonna be able to develop correctly, and we'll talk about why that might happen, or the adult, ultimately that's produced in the process of this hybridization, is sterile and unable to produce functional gametes to reproduce. So this all happens after the zygote forms. Any organism that's using a post-zygotic mechanism for reproductive isolation gets through those first two steps. They actually do make their zygote. So technically, they actually do make a hybrid. A hybrid would be an organism that's produced through the mating process of two different species. We get a hybrid zygote. Okay, so now we're going to walk through some of the specific examples of reproductive isolation mechanisms, and I'm going to tell you which of the categories they fall in. We're going to start first with our pre-mating isolation mechanisms. So whatever this process is, it's going to prevent mating from being able to occur between two individuals that are different species. The first way this can happen is through something called temporal isolation. When you hear the word temporal, I want you to think of time. This is going to have something to do with time. In this case, what's happening is you've got these two individuals that are different species, and there's a difference in when they are actively mating, and therefore they never mate with each other. Now, temporal isolation could be seasonal, meaning that you have different species that are mating at different times of the year. As an example, since we're in the spring semester, it's still pretty cold right now, when the temperatures do start to warm up, one of the first things that you're going to see coming out and blooming that's ready to reproduce is going to be our daffodils. Not buttercups. Some people call it buttercups, daffodils. Right? This is going to be a different group that we're going to talk about later. Right? So this is going to be one of the first things that comes out. This plant right here, tulips, closely related. They're not still in the same family. They used to be classified in the same family, but they are very closely related in terms of their families. But this one comes out much later in the season. So by the time these flowers are opening, even if they potentially could pollinate this, if you brought the pollen together physically and put them together with this flower, it might produce something. They can't ever do that because by the time these are sending the pollen out, these are already in fruit and they're done reproducing. They have no flowers left that can accept pollen, and so they're never gonna reproduce with each other. That would be a seasonal temporal isolation. But you can also have diurnal. Diurnal means that there are differences in the time of day that they mate. You've probably heard of morning glories before. They're called that because the flowers close at night and they open up in the morning because they're going to be attracting pollinators that are active during the daytime. And then there are other flowers that actually only open at night but are closed during the day. There are some things in the nightshade family that do that, which is why they're called the nightshade family. So if your flower is open and pollinators are coming to this, when this flower is closed during the daytime, even if a pollinator tried to come over here to this flower, it's not accepting any pollen because it's closed up. And the same thing applies at nighttime. It would just be reversed in that case. So they can't mate with each other because of temporal differences in when they mate. Also pre-mating would be something called floral isolation. But floral isolation can work in several different ways. There's actually going to be two different types of floral isolation. Just like there's two types of temporal, diurnal and seasonal, there's two different types of floral isolation too. One of them is going to be dependent on the behaviors of the pollinator, and the other one is going to be dependent on the structure and the anatomy of the flower. Okay? So the first type of floral isolation I'm going to talk about is something called behavioral floral isolation. Now this is going to happen because different pollinators, and I'm not necessarily talking about bees versus butterflies versus moths versus hummingbirds, even within a group, within the bees, you have different species of bees, you have different species of butterflies. Even within groups of pollinators, different pollinators recognize and see and sense the world in different ways and are attracted to different things. Right? Maybe they can see in the UV spectrum, maybe this one can't. Maybe this one likes this certain coloration or this position of the flower, maybe this one likes this one, this likes the scent instead. You've got different types of floral signals, colors and fragrances that these pollinators are specifically attracted to. And there's a lot of good examples of some different types of plant species that use this type of isolation. But one of the best ones are orchids. We're going to find out in the flowering families, that's one of the biggest families out there. There's about 20,000 species of orchids. And that means many of them are very closely related to each other. And the goal is not typically to try and hybridize. You don't generally want to fertilize or pollinate an individual that's a different species. Right? You want to pollinate or fertilize an individual that's the same species as you so that you know your offspring are going to be fit. Sometimes hybrids are not fit in terms of their ability to reproduce. And so the orchids are pretty specialized in their pollinators. And they oftentimes will have specific species of bees that they use as their pollinator. So you can see here the Orpheus fusa here specifically attracts Andrena bees, right? Whereas this species over here, Sculopax, is going to be attracting Eucera bees. A Eucera bee is never going to go to this flower right here. And an Andrena bee is never going to go to this one right here. The reason for that is the scent that they give off. The chemicals that are released by these flowers actually imitate sex pheromones. So they're actually specifically attracting male bees. And the male bees are coming to the flowers with the assumption that this is a female bee that they can mate with because that's what it smells like to them. And oftentimes the flowers are shaped that way, very similar to what the shape of the bee would look like. They're textured and they feel that way. You can see it actually trying to copulate with the flower. And in the process of doing that, it's getting pollen all over him. Okay? And then when he goes to another flower, which is going to be a flower of the same species because it's not going to be attracted to these sex pheromones, it's going to be attracted to these, it's going to pollinate the next flower in the process. Okay? All very specialized to try and prevent that process of hybridization. But it's dependent on these different bees having these different behaviors and being attracted to different things. So that would be behavioral isolation within the category of floral isolation. Now the other type of floral isolation is structural isolation. And this is specifically differences in the anatomy or the structure of the flower that causes different pollinators to pollinate the different species. It could be a situation where that pollinator is still attracted to that flower, but something about the anatomy of it, the position of it, okay, is preventing it from being successful in actually interacting with that flower. So here I'll just use an example. I've got two different types of columbines here. These are Aquilea, that's the genus name. We have one, the Formosa one, that's a flower that actually nods, which means instead of kind of pointing upwards towards the sky, which most flowers do, it's tilted downward. It looks like it's kind of bowing its head and it's facing downward. So the top of the flower is actually technically down here. This is the back side of it. Right? This one over here, the Aquilingia pubescens, is an erect flower, so it points upward the way we normally expect flowers to do that. You'll also notice on this one right here, on the back side of the petals, there are these little appendages right here that are referred to as spurs. That's actually where the nectar is, right down in the bottom of these tubes right here. So if you're a pollinator that wants to be able to access that nectar, you have to have some sort of a mouth part that can go down into that spur and reach that. Right? So you can see this one is a shorter tube, and the spurs over on this side are going to be longer tubes. So you're going to have a longer mouth part for that. These flowers right here, are specifically pollinated by hummingbirds. Right? Hummingbirds don't actually have to land on a flower to pollinate it. They can actually hover in place. They can be upside down underneath it right here and then have a long enough beak that they can get down into these spurs right here. But hawk moths actually have to land on the flower. A lot of pollinators do. They can't do anything or interact with the flower unless they can actually land on it. It just depends on the pollinator. But if they do that, they can't do that on a flower that's upside down. They'd be on the wrong side of the flower if they did that. So this one has to land on the top side. This one has to be facing upward. And the hawk moth that pollinates this has a much longer mouth part than a hummingbird has. So only that hawk moth is going to be able to pollinate this species, okay? And not vice versa, not the other one. It's not the fact that the hawk moth wouldn't be attracted to this. They're the same color, right? It's just that structurally, even if it tries to interact with the flower, it's not going to be successful. Right? So it's not a behavior situation. It's a structural isolation. Another good example of this are milkweeds. Okay? Milkweeds kind of package their pollen in an unusual way. It's going to be the same way that orchids do, and we're going to talk about that later in the semester. They produce these things called pollinia. And essentially what this is you're going to have these big wax balls of pollen all grouped together into one structure. So this is not one pollen grain right here. This is lots and lots of pollen grains all together in this waxy mass that you see right here. And there's two of them on both sides of this thing that looks kind of like a wishbone. Okay? This whole thing's called a pollinia. And instead of individual pollen grains being transferred by the bees or the pollinator, this whole thing actually gets stuck to it, and the whole thing comes, right? But each pollinia from each species, so you can see here's a different species pollinia right here, is shaped differently. It is structured differently. And in order for the pollen to actually get to the stigma and actually fertilize and mate with the flower, it has to fit in a slit right here, what we call a stigmatic slit on the flower first. Right? But this is kind of going to work like a key. Right? You have to have the right key. It has to be shaped the right way for it to fit inside of this lock. And if it doesn't, it's not going to go in, and pollination is not going to happen. It works a little bit like one type of mechanism that we talk about for animals, reproductive mechanisms called mechanical isolation, where you have male reproductive parts and you have female reproductive parts, and they should fit together like a lock and key, and if they don't, mating is not going to be successful. Same thing works here for plants. It's just kind of a different version of it, and we call it structural isolation under floral isolation for that reason. So different species are not going to be able to pollinate each other because they're not going to have the right shaped pollinia to do that. All right, now keep in mind, both of these two things I've just described here are types of floral isolation. So those adaptations can work either through the effects of the pollinator or through the structure of the flower, but they're both the same type or category of pre-mating isolation mechanism, which is floral isolation. The next one is that we're going to talk about is going to be a type of post-mating prezygotic isolation mechanism. So before we can get into that, we need to understand a little bit first about what exactly happens if mating is successful in a flowering plant. Okay? What's going to actually happen? So let's say you do manage to get this pollen grain, right, finally to the female reproductive part of a flower that you're trying to get it to. What happens next? And once that pollen grain gets there? Well, here we have our female reproductive structure. Here's our ovary, here's our style, here's our stigma up top. Stigma did its job. It caught the pollen grain. There's the pollen grain right there. Next step that has to happen, now that it has the pollen grain and mating has been successful, is it will start to grow what's called a pollen tube. So the pollen grain is going to produce this tube that is going to go all the way down here, all the way to the bottom of the ovary right here, and actually penetrate one of these two blue cells right here. It doesn't matter which one it does. And it's going to eventually, once it gets down here, release two sperm cells. We'll get into later why there's two. instead of one, but there's two, and they're both involved in the fertilization process. And it wants to get to here, this point right here, which is the egg, and that's what it's ultimately going to fertilize, if it's successful, okay? But remember, this is still gonna be a prezygotic isolation mechanism, which means we, for some reason, are not making it all the way to the point here where the sperm is actually managing to fuse with the egg. We've managed to get the pollen grain there, but why is the sperm not getting to the egg? Okay? That's gonna be a type of isolation mechanism called pollen pistil incompatibility. It's gonna involve the style and the stigma of the female reproductive structures that are involved, right, in this particular mating. And what happens here is that there's a chemical reaction that's occurring. There's chemistry at work here. The style and stigma, right, are actually preventing potentially the pollen tube from growing. Okay? Some flowers can actually choose which sperm they want to accept and which they can't. Not all, but there are some species that can actually choose which sperm they want to accept and this is not something they want, the pollen grain they want, they can just stop that pollen tube from growing, and if it can't grow the tube down to the base of the ovary, it can't fertilize the egg. So that would be all based on chemistry. But there's also something else that can be at work here. Let's say that the pollen tube is going to grow, right? Something's going to get there at some point. The question is, which one's going to make it? Let's say, for example, you have two pollen grains that have landed on this stigma, right, essentially at the same time. One is from the same species, right? One is from a different species. Okay? And in this particular case, the plant wants the sperm from the individual that's the same species. Right? So what's gonna happen here is it can actually control which pollen tube grows the fastest. Make it easier for the one that it wants to get down there and slow down the one that it doesn't want. Because it is a race to the egg. Who gets there first? Whichever sperm gets there first is the one that wins, that actually gets to fertilize the egg. So if it's the slowest pollen tube, it's not gonna be able to deliver the sperm fast enough to do that. So same species pollen tubes may grow more quickly because of the chemistry of the style and stigma in same species styles than different species pollen tubes. Okay? You can also see there's some differences here in lengths of pollen tubes. If this distance it has to travel is too long for the pollen tube to grow, that's also a possibility in terms of stopping that fertilization as well. So we've got post-mating prezygotic isolation mechanism, because we're still not making a zygote. We've made it, we've managed to do that, but we can't do fertilization. Okay, now, let's say it is successful. We're gonna get into post-zygotic isolation mechanisms now. We've managed to get the pollen tube all the way down here to the egg. We're gonna deliver the sperm, where the sperm is actually going to fuse with the egg. We're gonna get a zygote, a hybrid zygote. What then can happen beyond that point? Well, the next step in the process is that the organism is supposed to be able to develop. And that's something that potentially could be inhibited. It could be stopped. You have something called seed incompatibility. Seed incompatibility means you get your hybrid zygote. You get that cell when the sperm and the egg actually fuse together. You may even get the next step beyond that, which is called an embryo, which is a multicellular stage that's just beyond the zygote, but you're not quite at the adult yet. Okay? You may even get to that, but it's not going to get much further than that. And in most cases, in this situation, you can't even develop a viable seed. Now, why might that happen? Most likely what's happening is that there's some sort of incompatibility between the parental genomes. You have to think about what's happening here. When you get a sperm, when you get an egg, you're getting directions, sets of directions, DNA, from both parents. Right? It's a very important part of the reproductive process, is making sure that the genetic information that you have gets transmitted to the offspring. The reason that's important is because they need to have a set of instructions on how to build themselves and function properly as well. So you have to pass that on. But here's the problem. You have a male that's one species, a female that's another species. They develop in different ways. They have a different set of blueprints, a different set of directions. So let's say, for example, you're getting a set of blueprints for making a roundhouse, and the mom is giving you a set of instructions that's making blueprints for a square house, and you're supposed to be making something that's some combination of both of those that's functional. What's the likelihood it's functional? Probably not very likely, right? Because if you start putting square things on in the roundhouse, it's not going to work properly. It's not going to be stable. It's not going to be able to hold itself up. That happens. If you've got two different sets of directions, and the male's telling you to develop this way, and the female ones are telling you to develop this way, you're going to get something that's not right. and not gonna be functional. So you may not get a seed at all. You could also have hybrid inviability. Now this is where you actually do get a hybrid. The development process is starting. You get much further than you do for the seed. You may even get a seedling in the process of this, but they don't survive long enough to reach maturity. Now there's multiple reasons why that can happen. One of the most common reasons this might occur is if you have different environments that are required for the parents. So say for example, the male species was a plant that's adapted for living in very wet conditions. Now that's the habitat that it's in. There's a wet habitat right here. But it's right next to a dry area. And some of those pollen grains actually made it over into this other habitat and pollinated a different species of plant that lives over here, meaning that the mother is adapted for very dry conditions. Well, their offspring that's produced must be a combination of the two parents. Maybe something that's not adapted either for wet conditions or dry conditions. Maybe more mesic conditions, something in the middle. But that habitat doesn't exist. There's nowhere around in that area. You have wet or dry, that's your only option. And they can't live in that habitat because they're a combination of these two parents. So they don't survive long enough for them to ever eventually reach adulthood. But you could also get hybrid floral isolation. Now, this is going to oftentimes be mixed up with floral isolation. It is related to it in some ways because it is going to deal with the structure of the flower and pollinators, and in some cases their behaviors. But this is different than the original floral isolation I taught you because that one happened before we actually got to the zygote. This one happens afterward. This is relating to that actual hybrid itself. So you've managed to get the two parents to mate with each other. They've managed to produce a zygote, a seed that develops into an offspring. And let's say this individual actually even makes it kind of up to adulthood in some ways. But the problem is the parent plants were adapted for very different pollinators. Let's say the male plant was adapted for a bee pollinator and the female was adapted for a hummingbird pollinator. So they had certain flower structures that were better suited for each of those two different types of pollinators. When you get the hybrid flower is some combination thereof, which means structurally it's probably not good for either the hummingbird or the bee, right? Neither one of those pollinators can actually pollinate them. But one of the conditions of the biological species concept says that offspring has to be able to reproduce itself. If it can't have a viable or can't have a pollinator, can't find an effective pollinator for it, it can't do that. In which case these two parents would, by that definition, be considered different species. So that's hybrid floral isolation. All right. The last one here that we're going to talk about from today, we'll get into the next time, is something called hybrid sterility. This means that you make it pretty much all the way. You make it through mating, you make it through fertilization, you make it through development. They're adults, they're robust, they're healthy, right? Everything about them you would think was perfectly normal, and this has been a successful breeding operation. The problem is the offspring that's being produced is sterile. They're not going to be able to produce functional gametes. So as much as they want to reproduce with each other, they're not going to be able to do that. It's the same thing that happens when you cross a horse with a donkey. You get a mule, right? They're healthy, they can grow to adulthood, they can live long lives, they're very strong, but they are sterile. A mule cannot mate with another mule. It's just not possible for them to do that because they have non-functional gametes. Works the same way here. Cross a brassica here with a rhapidus, or a rhapidano, brassica. You got a nice little radish-looking plant right there. Leaves are healthy, can photosynthesize, it's got roots with water nutrients. It's sterile, so it can't reproduce. Hence the hybrid sterility. Okay? All right, we'll get into hybrid breakdown in the next class.